Hello, everyone. My name is Brandon Ching,
Hi, my name is Chelsea Tafoya.
and today we're going to be talking to you about the research that we have been working on these past few months regarding the identification and characterization of genes from phage Amelie that are toxic to Mycobacterium smegmatis. So as I'm sure most of you know, the mechanism by which bacteriophage are able to infect their bacterial host is quite complicated. Not a whole lot is known about the subject quite yet. With this in mind, Chelsea and I decided to focus our research on shedding some light on this mechanism of interaction between a phage and its host. In order to do so, we began working with a K1 bacteriophage known as Amelie and aimed to further characterize its genome through multiple assays. The first assay we went through is known as the cytotoxicity assay and tells us whether or not the gene products from Amelie are toxic to our host bacteria. The second assay is known as the defense assay and tells us whether or not our gene products are able to provide defense against infection from other phage. The third assay is known as the two-hybrid interaction assay and allows us to determine which bacterial protein our phage protein interacts with. So diving straight into methods, here's a figure that illustrates how exactly this assay works. So put simply, in order for growth to appear on our plates, we need to have some kind of interaction between our phage protein of interest and protein from the host proteome. Both of these proteins have been fused with transcription factors that work to regulate transcription of two genes that are necessary for cell growth on our medium. In order to scan MSMEG's whole proteome, a plasmid library was constructed by our collaborators over at HHMI. This plasmid is pictured here and labeled PCI, along with the plasmid that we created in the lab containing our phage gene of interest that's labeled P2H alpha. Now, once both of these plasmids are transformed into our host, the growth of colonies tells us whether or not an interaction between host and viral protein has occurred. If there is interaction, the transcription factors are able to work together to recruit RNA polymerase and transcribe the genes that provide our host with antibiotic resistance, as well as a reporter gene that will turn our colonies blue. If no interaction occurs, then the antibiotic resistance will not be provided, and we will see no growth on our plates. So after this process, if we do see growth on our plates, we must go through several procedures to confirm that they are true hits and not false positives. And once we are certain that it is a true hit, we then perform a sequencing reaction and send it to our collaborators to be sequenced. Pictured below that is a chromatograph, which is kind of an example of one of the sequences that we received back from our collaborator. And this shows us the protein that is able to interact with Amelie GP47. Interestingly enough, GP47 is also able to interact with another protein from M. smegmatis' proteome known as NUS-A. This figure is a map of Amelie's whole genome and works to visualize the results from our cytotoxicity assay. As you can see, the genes with a white circle above them are toxic to our host bacteria, whereas the genes with a black circle are non-toxic. Now, below the map here, we have the results from our two-hybrid interaction assay when we performed it on GP47. Now, each plate contains different concentrations of a recovery mix that we plated. And as you can see, they each have some kind of colony growth on them with blue colonies. And this tells us that we have a true hit and that there was indeed an interaction between GP47 and our host bacteria protein.
Additional results we gathered were for the sequencing reactions for all the genes we managed to clone into the P extra plasmid. We were able to clone 76 out of the 77 genes. We were able to confirm all 76 out of the 77 via PCR and gel electrophoresis. Out of the 77 genes, we were able to successfully verify 74 of those genes via sequencing. In total, we were able to confirm 95% of the plasmids via sequencing. Additionally, we were able to perform interaction assays for 5 genes. As stated before, we found that GP47 of Amelie was able to interact with both the transcription factor NOS-A as well as the bacterial protein PRNT. GP47 is part of a family of genes found in clusters K1, K3, K5, and K6 phages. Interestingly, GP47 of phage waterfowl, which is homologous to GP47 of Amelie, was only able to interact with NOS-A. We were able to perform defense assays for 14 genes, but so far we do not have any significant results. Some further experiments we would like to do include identifying regions of interaction with Amelie GP47 and M. smegmatis proteins NOS-A and PRNT. We would also like to sequence other cloned genes and identify their interaction partners. Lastly, we would like to further characterize mechanisms of action during infection. Thank you.